So now we're going to take a little bit more of a dive into concepts of species interaction. So I mentioned previously that species and their interactions through niche theory and through things like competition drive a lot of the distributions that we see for plant life. And I want to quickly refresh also that concept of ecosystem, which again is this pairing of those non-living elements, the abiotic factors, with our biotic factors. And of course, when we think about that alchemy of how these will interact, especially when it comes to our species, what we're going to see is that distribution, number of species, the time during which that species arrived in a habitat will have a great influence on what that ecosystem will evolve to look like. So we've already talked about competition, which is that rivalry for common resources. When we talk about competition between individuals of the same species, we call that intra or interspecific, like introvert. So within species competition, talking about competition between species, we call it intra-specific, like international. And then, excuse me, I have that backwards. Intra like introvert, inter like international. And then something else we kind of mentioned before in the chat afterwards was facilitation. So that's where one organism is benefiting another organism and allowing for that organism to grow, but with no benefit to the organism that's providing that benefit. And in a non-plant world, that's often referred to as commensalism. And then mutualism is where there's kind of a two-way interaction where each species is getting something out of that exchange. And we refer to mutually beneficial interactions where each species is benefiting. A lot of times in lay terms, that's called symbiosis. But it's important to note that symbiosis is just, again, an interaction between species. It doesn't say exactly what the outcome is for that interaction. And then to be plant specific, we should talk about herbivory, which is where some organism is munching on our plant. And sometimes people like to associate parasitism, so a parasite, as being a form of herbivory. I think that one distinction there is, though, usually with parasitism, the parasite is not only munching on the plant and taking resources from that plant without killing it, but also it's using that organism as a home. And then predation would be where one species is killing another species. In this case, with plants, we might be thinking about an organism where maybe it's eating a bulb or eating a seedling and therefore killing a young plant. So that would be an example of predation. Worth knowing that predation and competition are different. So even while I think sometimes people will mistake those two, remember that competition is for a common resource, whereas predation is one organism is the resource. There's also some other interactions and paradigms within ecology that I want to touch on, some big picture things that organisms can play within the species or habitat in which they interact. Those are our dominant species, our keystone species, and our ecosystem engineers. So dominant species are organisms like a redwood tree that have a defining role within a habitat. A redwood tree is obviously the defining attribute of a redwood forest, and that redwood tree provides and creates habitat under which many understory species will be able to grow and thrive. But it's worth noting that that dominant species, its impact in that system is relatively proportional to its physical size in that space. And that's different from something like a keystone species, where that organism has an outsized impact relative to its biomass. So an example of a keystone species might be salmon. And salmon in our northern coastal forests, such as here on the Smith River in Delaware County, play a pivotal role in bringing nutrients to these systems. So the Smith River is highly oligotrophic, meaning nutrient poor. It receives a lot of rain. Even in the wintertime, the water appears crystal blue. And what happens is these salmon, they're reared in the river and tributaries of that river. And as they grow and mature, they swim out into the open ocean. They feed on nutrients that are rich in the ocean. And then at a certain point, they get this sensation to return home. And as they return home back to their ancestral waterways, they spawn, so they procreate, and then they die, and their carcasses are left on the riverbanks, which are then consumed by other organisms and then distributed as waste throughout the forest. And of course, that salmon body, if you eat salmon, you know they're rich in protein, which means they're rich in nitrogen. And so a forest that's starved of nitrogen ends up relying on things like salmon to deliver that nutrient into that system and keep that system healthy. So in this way, the salmon, diminutive in size, has this large impact in the health of that forest by bringing nutrients into that system. And it helps to keep that system stable. Ecosystem engineers are a little bit different. They also have an outsized impact relative to their biomass. But here, rather than maintaining a status quo of our system, now that organism is driving a change in what that system looks like. So we can think of things in the Fabaceae family, such as lupin. That's the pea family, which have nitrogen fixing microbes that they associate with. Those nitrogen fixing microbes change the composition of the soil, and in changing that composition of the soil can drive that system in a different direction, can lead to other organisms, other plants being able to establish themselves in that space because now there's a resource available that wasn't available before. So here, rather than keeping something in a status quo, we're changing that system. And a common example that's a non-plant example would be a beaver. Beavers will build dams. They will change the flow of water. They'll raise the water table in a system and allow for a different water table that can affect different plant life and different patterns of the stream ecology as well. We also want to consider density again, and also to think about priority, which is time when something arrives in a system. So this is an example of an alder forest. Alders are often associated again as being nitrogen fixers and also being sort of nurse species that allow for redwood forests and other conifer forests to fill in at later times. But you can see that in sometimes very dense stands of alder, it may be that that density prevents establishment of conifer forests, at least in the interim. And also when we have high density forests of alder, even while that forest may be facilitating, so providing for growth of conifers, providing for other species to fill in, even while that starts as facilitation, it may ultimately become competition as those conifers replace and shade out the alders, which will ultimately die and leave that system when the conifers now have affected a system that's too shady and too dark for the alders to flourish. So time and also density become really important in thinking about the effects that organisms will have in their interactions. And this leads us to a concept called succession, which is how a landscape changes as species grow and are introduced to that space. And I think the most broad brushstroke terms, we can think of succession as a move from more ephemeral landscapes to more sturdy, robust systems. So that could be the replacement of annual grassland systems with forest. And this is a picture from Point Reyes where you can see encroachment by douglas-fir onto grassland systems, which are changing that from a grassland habitat to a fir forest. So early ecologists in Western ecology thinking saw succession as essentially a one-way movement where you went from these more ephemeral spaces to more complex, robust and resilient systems like forests. What we know now is that in places especially like California, it's much more of a conveyor belt. And so we don't go to what was called a climax community. Instead, we have a rotation of systems, and that these systems emerge depending on various disturbance factors like fire or herbivory or wind throw that can lead to the removal of some aspect, allowing for another ecosystem to flourish. And this produces a mosaic of different habitat types, which fuels a lot of the biodiversity that we see in California because of that high turnover of our landscape features. This is important to think about as well, because when we have systems that are out of balance, we can see that that has an interruption in succession or an interruption in what species will be dominant in a system, what species will thrive, and then which ones will ultimately lose out. So I want us then to think about invasive species to change track a little bit. And this idea of invasiveness and an archetype for what invasive species look like in a system was first considered by Baker. Herbert Baker worked at the Jepson Herbarium and UC Berkeley for about 44 years. And during that time, invasiveness and what caused invasiveness were some of the central things that he focused his work on. And I was trying to reduce his first paper into a couple key pithy points. And I realized that as I was doing that, they, if we're arranged right, made this acronym, which is GRIEF. And invasive species will give you a lot of grief if you don't really understand the mechanisms that allow them to succeed and allow them to prosper on a landscape. So I think an important attribute that Baker identified is that invasive species tend to be generalists. And we mean by generalists, we're saying that they are organisms that are gourmands, not gourmets. They will happily inhabit almost any ecosystem type that they arrive in, and they do very well without being specialized to a particular landscape feature or attribute. And that kind of makes sense that if you didn't have that trait, we wouldn't expect to see them being the rabid colonizers that invasive species typically are. We can also speak about them then being easily dispersed and having easy dispersal mechanisms. So this is a pampas grass pictured in this image here, which comes from the Andes. Pampas grasses are rampant throughout much of the California Coast Range, and they make these beautiful, fluffy, feathery reproductive structures with seeds that are very easily, when born, that very easily stick to clothing and can be moved great distances. And again, that fits very well with the generalist idea in that this is a seed that's designed to go far, to be able to colonize many different spaces. And then on that same note, easy reproduction. So we might think of that in two ways. We could think again of this pampas grass, which is producing thousands of seeds, maybe even tens of thousands of seeds in its lifetime, we might expect, easily able to disperse. But also, in addition to sexual reproduction, we can also think of asexual reproduction. So if an invasive shows up in a habitat where there are no other individuals of that same species, being able to reproduce vegetatively, so from cuttings, from tissue, can also be an important strategy to spreading on that landscape. And we see a lot of invasives in California, like Cape ivy and Cape weed, that will actually spread predominantly through bits of tissue that have broken off and spread in these vegetative manners. That can also be beneficial because vegetative structures tend to be easier in terms of resource acquisition for reproduction, less energy intensive as opposed to producing a flower and producing fruit. And then also something else that comes to mind when we think about that easier mode of reproduction through vegetative structures is that if you're a plant that can easily grow from a little bit of vegetation, then it also means that you're a plant that can easily recover if you're destroyed or knocked down or broken up. And so again, that speaks to a certain hardiness in our plant. And as I mentioned before with our alder stand and rate of growing and rate of colonization, priority when you show up in that landscape will also be important. So many invasive species, these species that have essentially been rabid in their colonization of habitat, often are very good at taking habitats that have been disturbed. So maybe a clear cut or the area after a landslide or some area that was recently burned, very easily at filling in and growing into those spaces. So that's another attribute then important when thinking of invasiveness. And then lastly, introduced. And this kind of is two directions I want to take this. On the one hand, we can think of introduced meaning species that are not from California, that didn't adapt to this landscape. And when we speak of introduced species in that context, we're thinking of things that have what we call novelty. And sometimes we call that the novel weapons hypothesis, which means that these are species that are bringing with them traits that allow them maybe to have an advantage in that system. Maybe it's a toxin, maybe there's no herbivore to keep them in check or a parasite that would reduce their spread. But we can also think of introduced species that are native within their range. And an example that comes to mind for me, to go back to that slide that showed succession, is douglas-fir and also a plant that grows along the coast that I study, which is Bacchus pilaris. And both of these plants are wonderful species. I enjoy each of them. They have a lot of great habitat benefits. But because there has been a removal of certain factors in the environment, so an introduction of a human element that's prevented fire and herbivory from keeping these species in check, we can see that they spread prolifically through their native range and can act then as an invasive element within a habitat. So because of this, when we say invasive, we don't necessarily only mean non-native species. We can also be talking about native elements as well. And if you're interested in learning more about and identifying native species, I've included a link to CALIPSI, California Invasive Plant Council website that has a watch list of species that are out of place. But I think it's also important to have an ability to identify species that may be either invasive or prone to invasiveness. When I worked in restoration, folks would often stop me and ask me if the species that they were looking at or a species in their backyard was invasive or worth removing. And of course, that depends on your objective in the landscapes you want to manage. But at the same time, I think using Baker's criteria is a really important stepping stone for gauging whether or not something is going to have the attributes that will make it invasive or something that you should be watching out for. And that goes a lot, I think, to show that also there are many species that are non-native that may not have a negative impact on the landscape. when I worked in the Redwood Creek watershed, I often saw things like Thai basil or tomato plants that were growing in the creek channel that had washed down from this little inholding of a farm upstream. Those plants, they only lasted for about a season. They weren't prolific. They didn't spread. They weren't really adapted to that space. They didn't have any of those traits that Baker outlined in terms of being aggressive in their colonization, being generalists, or having a novel attribute that gave them a leg up in that system. So in thinking of ecological diversity, I want us to formally define what we mean by diversity. And essentially we're bisecting that into two terms, which are richness and abundance. And we need to have a concept of richness and abundance and ecological diversity if we want to think about spread of invasives. We want to think about the growth or proliferation of some native species of concern to us. So in thinking of ecological diversity and richness implies all the different species that we find present in an area that we are surveying. And then abundance would be, and in particular, relative abundance would be the proportion of those different species in that area. And thinking about proportionality is important because you can imagine that if there is, say, a really small quantity of some species present in an area, that species may not be functionally able to reproduce or spread and may be at risk of maybe a local extinction or retreat in its range, or may not functionally be able to aid in a pollinator or recovery of some other species and play that niche role that it has within that system. So there are ways that we can measure diversity, and I want to overview some very simplistic methods for that. So one approach that we can take, if say we were to survey this spread of understory redwood species, will be with a line transect. So we can just run out a tape measure and then count all of the species that intersect that line. So in this case, we're going to count all of the individual plants. And some of these species, I'll note, do have runners that mean that they may be growing together under the duff that these plants are growing from. But for this analogy, we'll just imagine that we saw, when we counted up, 4 of these oxalis, which are those clover-shaped leaf plants, 2 of these asarum, which is wild ginger, and then maybe 1 of these gallium. And so if we were to think about richness of this habitat, we could say that there were these 3 different species present. And then in terms of relative abundance, we would be taking a percentage of those species represented. So we might say that we have 4 of those of the oxalis from 7 total individuals that were counted, and that would give us then 57% of that population that we surveyed, as of that community that we surveyed, as being the oxalis. So that would be how a line transect would work. And this is really effective when we're dealing with smaller organisms that are close growing together, like grassland systems, understory species. But we can also imagine situations where we may have species of different sizes that are spread out. So maybe we want to survey these species, but also a redwood tree or other tree that's in close proximity. Or maybe we're working in a desert habitat where these species are all very spread out, and we may not be able to encapsulate those species along our line transect. So here we can do what's called a strip transect, now expanding the width of that transect. Again, not necessarily as applicable to a system like this where things are close together, but you can see just from this example that we've added another gallium to our abundance count. And so it's worth knowing that however you go about surveying an area of land, it's important to be consistent in your approach and to also make consideration of how that survey method will best encapsulate the diversity that you're trying to measure. And also, in terms of a statistical approach, to survey as much as you can, because that way you can correct for any errors that may exist in that data. But there's something else in terms of size and comparison of species that we need to consider. And so for that, let's go even more to a 3-dimensional perspective with a quadrat. And this quadrat here is kind of a throwback to what we looked at before when we were thinking about species area curve in that first chapter. A quadrat could be a 1 by 1 meter area. It could be a hula hoop, whatever you see fit or have available to you. Here, if we're looking at a quadrat within that 2-dimensional space, we might say that there's 2 organisms represented, right? So richness of 2, and then abundance by individual, we could say 1 asarum, 1 oxalis, right? But this doesn't really encapsulate the actual space that's being taken up by these organisms. So here, instead, we might think about the spatial abundance of those species present. And so rather than thinking about this in terms of individual count, we might say, all right, let's look at the asarum. We see that they take up the most area, maybe 75%, and we've got perhaps 14% that's oxalis, and maybe 16% that's our soil. I don't know if those values are up to 100. It's okay if they don't, because we have to recognize as well that there's a three-dimensional aspect. So there may be a serum growing underneath some of that oxalis, or oxalis growing on top of some of that asarum. But ultimately, this gives us a cleaner image of biomass that's present within that system. This is really important if you're trying to compare, say, abundance of a grass versus abundance of an oak tree, because you can imagine that 30, 50, 40 individual Stipropulchra, which is a species of grass, will not in any way compare to the biomass of a single mature oak tree. So when taking all of these survey methods, we can then use them across time to create comparison points and create benchmarks to monitor how systems change, how succession is having an effect, how the spread of an invasive species is influencing a system, or how our restoration work is allowing us to create a system that has sustainability and functionality, and will ultimately influence, I think, our management decisions, and also what we can affect in terms of long-term goals for protecting our natural spaces. And I've included CNPS field protocols, as well as other monitoring protocols set up by USDA that give greater depth into some of the approaches you can use if you decide you'd like to do some surveying of native abundance in your area, as well as links to Calypsy's invasive plant weeds to watch out for list. All right, I'll turn it over to you, Amy, for questions.